Thank you very much for coming today, and I would first like to thank the organizers for giving me the opportunity to present to you some things we can offer to you from Fresenius Kabi. So you heard now about drug delivery, more or less from all dosage forms, and I would like to enlarge this a little bit and give you an overview about IV forms of drug delivery. So this is the drug delivery, more or less the technical aspects of IV forms for this. Because drug delivery has, from my point of view, several meanings in the pharmaceutical community. There's first a technical side, which I want to focus on. So galenic formulations of drug molecules. There, of course, the common goal is to enhance the efficacy of drug molecules. And also one aspect is the transport of the drug molecules to the point of their actions. But as already mentioned a few times, this presentation will focus more on the technological aspects, presenting also our capabilities in Fresenius Kabi. So therefore, I would like to give you a short overview about Fresenius, which some of you might not be aware of, because we are listed as a healthcare company, not so much as a pharmaceutical company. The Fresenius is SE, has a sales of around 16 billion, so it's quite a big company already with 150,000 employees. Biggest part is Fresenius Medical Care, which is dedicated to dialysis. I'm coming from the Fresenius Kabi moiety, where we deal more or less with pharmaceutical products. We have around 25,000 employees currently worldwide, and we are really focusing on hospital products for chronically and critically ill patients. So we have several therapy areas that we are working on. This is the infusion therapy, nutrition therapy, enteral and parenteral nutrition, medical and technical products, and of course some areas that are most interesting for me, because I'm responsible for that, for the third-party manufacturing, which starts with the API business, where we can offer beta-lactam antibiotics, oncology drugs, starches, lactulose, oils, phospholipids. And then we have the third-party manufacturing for medicinal products and for medical devices. All these groups that are responsible for that you can meet at this short Hall A4A44. This is the booth number. As you can see, we have manufacturing sites all around the world, with a slight focus on Europe still, because Fresenius is a German company, but we can deliver to all over the world and from all over the world. My sales team is, as you can see, can be found at 4A44. Sorry, this is a little bit this. And you can find my colleagues, sorry. For the business development, all the directors of contract manufacturing can be found there, and will be really happy to discuss with you any needs that you might have. So, coming back to drug delivery, which is currently a quite big business. Can find in the literature some values. One I took, it's the 27% of all pharma sales in 2007, and already 32% this year. This can be found from IMS data that are available. And of course, every kind of drug delivery technology is included in these figures. Just a short summary of how we see the drug delivery in different aspects. As I already said, it's a galenic formulation of drug molecules. Focus is also to prolong the shelf life of a certain drug inside the body. Then we can play around with the route of administration. And finally, also drug targeting is a big topic in drug delivery. So as I said, I will later on focus on the first point here, but would like to give you a brief overview of all the other points here as well. Let's start with the prolongation of shelf life at the site or during the life of action of the drug. So all the drug carriers that we are preparing are really expected to increase the shelf life of the drug molecule inside the body, so to increase the time of action for this specific drug. There we can make different approaches. We can have controlled release for oral forms, for subcutaneous applications. I think my previous colleague here from Catalan gave you a very wonderful overview about the solid oral, solid dosage form possibilities, and I can only recommend that you visit them if you have some kinds of needs for this. We are more focused on the IV side of the drug molecule or the drug targeting. Then we can have chemical stabilization of the drugs. For example, pegylation might be very well known to you, but out of our company we offer hesylation, and there was a presentation yesterday in the afternoon about this topic, so I will not consider this because we have already talked about that. This is nearly the same as pegylation, but just replacing polyethylene glycol with hydroxyethyl starch. And we can also change the route of body clearance for a specific kind of drug. This is, for example, done when using a stealth liposome instead of a standard liposome, let's say. I will come back to this a little bit later. Then we have, of course, the route of administration. So this is also, if we change or alter the route of administration, we always try to enhance the effectiveness of the drug in the body. Also here are a lot of examples for this, and please be aware that all my examples here are certainly not exhaustive. So there are certainly several other technological means on the market, but this is the most common that I see. So for oral delivery, we have seen already that sustained and modified release is used for drug delivery, and we can use inhalation, intranasal and pulmonary delivery. Vaginal, rectal delivery is feasible. Transdermal, of course, using the biggest organ of our body, the skin, and topical route of administration. And again, I'm focusing then later on the injection route of administration here. Another aspect I mentioned earlier is the drug targeting. So the primary focus of this wording is, of course, to bring a specific drug as close to the site of action in the body as possible. So avoiding any negative side reactions in other parts of the body. Also here, people have had very creative ideas of how this can work. So there have been pH and temperature sensitive formulations on the market, well knowing that in areas of disease like cancer cells or inflammation or anything else, there might be microenvironments that have totally other environments than can be found in other parts of the body. So there can be a different pH, there can be a different temperature in these areas. And then these drugs should come in action and be released from their drug delivery vehicle. There have been some attempts with magnetic formulations, just applying a magnet from the outside and hoping that the drug delivery vehicles are accumulating in these areas where this magnet is applied. And of course, one very promising technique is an antibody coating, either of the drug itself, it's big enough, or of the delivery vehicle that is used for drug delivery. So just a short summary about these last slides. The primary focus of the drug delivery is, of course, to increase the effectiveness of a drug molecule and reduce unwanted negative side reactions in other parts of the bodies. And in our point of view, these targets can only be reached by big technological improvements. There we have two sides of actions. Either we start with a medical device where we can apply the drug molecule in a different way as it is used now, or we do use improved galenic formulations of the drugs. So, as I already said several times, I will focus on the IV route of administration of drugs, and so I would like to give a short overview of what is necessary to be able to administer a drug in an IV route. There are some more stringent requirements, I think, compared to oral solid dosage forms and other forms, because the drug must be sterile. It's not always easy to get sterile drugs, drug formulations. It must be free from visual particles, and it must be liquid, at least when it is used. Furthermore, it must have the right osmolarity, it must have the right pH for application, and in some cases, as we will see, also the right particle size. And standard forms for IV drug delivery is, of course, a ready-to-use formulation, which is the best if you can use that, because you don't have to handle it in the hospital again before use. And this is mainly comprised of solutions, aqueous solutions mainly, emulsions and suspensions. Then we can have powder products, where a powder is filled and before use it must be dissolved in this case. And a special form of powder products, of course, lyophilized products, which are first in a liquid form, then in a powder form, and finally again in a liquid form when you dissolve it before use. What kind of formulation techniques can we offer to you as customers to enable the delivery of several drug substances, drug molecules? We have the ability to formulate emulsions, liposomes, suspensions, and some chemical modification of molecules. This is specifically mentioned with the hesylation technology that we offer, but as I already said, this will not be part of this presentation here. So let's start with an emulsion. It's a very standard emulsion that is schematically shown here. We have a continuous phase, we have a droplet phase. Normally in standard form, the continuous phase is water, the droplet phase is a lipid, but it can also be the other way around. And we need some emulsifiers that show a hydrophilic head and a hydrophobic tail. And inside this droplet phase, inside the lipid phase, we can place a drug molecule that is quite hydrophobic in its nature. Sorry. So in principle, we have the lipid phase, which is more or less an oil. There are several oils available on the market. Emulsifiers, currently mainly lecithins, because they are most known. Some additives like glycerol to have the right osmolality in the continuous phase, and water as the outside. What advantages do we see in the emulsions? Well, they are very well characterized currently because they are used since decades, both for parenteral nutrition only. If you cannot eat anything, then you are really dependent on enteral or parenteral nutrition. In each case, you need lipids, as you know. The manufacturing process is already very well established. And all the excipients I mentioned previously, especially the lecithins and so on, they are very well characterized and really regarded as safe. A small disadvantage, of course, also comes with this form. You have, of course, an additional lipid intake into your body that you have to add to your normal calorie intake. And this is sometimes not really wanted, especially in a state of disease. What kind of examples do we have? For example, intralipid, which is used for parenteral nutrition, and as a drug delivery vehicle, we have a well-known anesthetic, which is propofol. Vitamin E is delivered via emulsions, and diazepam as well. And coming to liposomes, liposomal technology is a little bit more difficult than the emulsions because it consists only of phospholipids, which forms in this case not a single layer but a bilayer to be stabilized, and we have a continuous phase outside in the water and also the same phase inside a liposome. But this offers also a great advantage that we can use liposomes in different kinds of drug delivery vehicles. So as you can see, the The active substances can be enclosed inside just to shield them from other actions in the body, or if they are lipophilic, they can also be solubilized in the lipid bilayer that we have here at the liposomes. Then we can add several attachments to the liposome, like protective molecules for immune destructions, or homing peptides that are looking for a specific site in the body, like antibodies and so on. In principle, the liposomes consist of phospholipids, most cases of some additives to make them more stable, like cholesterol. A lot of modifications can be added to these liposomes, and of course, again, water. So what kind of advantages do we see? As I already mentioned, there are many possibilities to carry drug molecules and to modify the surface structure. Also, these excipients I mentioned here are very well known. Manufacturing processes are established. There are several, from high pressure homogenization over evaporation of organic solvents. So there are some available. You have here, in comparison to the emulsions, you have a very low lipid index, just lecithins, that's nothing in principle. And also these liposomes can be modified so that they are seem suitable to carry the DNA and RNA for any kind of aims that we have with these molecules. Disadvantages are also here. The chemical and physical stability of standard liposomes, I have shown them here, is very difficult to manage. It's very short-lived, so most often liposomes are lyophilized after manufacturing and have to be dissolved before usage. And standard liposomes have a very fast body clearance if you use them. So there has been a development ongoing called stealth liposomes, for example, which uses polyethylene glycol as a barrier from the outside so that the mode of action is much prolonged inside the body. We have also some examples on the market already. This is Ambisome amphotericin B, which is an example for a lyophilized formulation. And there is doxorubicin, which uses a stealth liposome as the way of transportation of this drug molecule. Finally, come the suspensions. This is for us also quite new technology. And this is more or less a dispersion of non-soluble drugs or other molecules in a liquid environment, mostly water in this case. There are several formulation technologies to keep this system stable, as you can imagine. And they need special handling, special filling processes that they do not accumulate and sediment during the filling process. There are a lot of examples out in the market, but they are limited for subcutaneous and intramuscular use, so they cannot be used IV directly in the vein. One of the examples is medroxyprogesterone with the product Depo-Provera, for example. The other one is that I would like to mention is dexamethasone under the brand name Decadron LA. Finally, I would like to show you some possibilities that we have where we can fill these formulations into. So we have a quite huge range of container forms that we can use to apply IV solutions. Of course, ampules, very widely used these days. We have from plastic ampules from 2 milliliter up to 30 milliliter, and glass ampules, approximately the same range. Then we use often IV bags for ready-to-use formulations. There we have a range from 50 to 1,000 and even larger milliliters up to 5 liters. And of course, everything that comes into glass. So glass bottles, glass vials, just a point of definition, I would say. And the same we can offer in plastic as well. So we have glass bottles up to 1 liter and plastic bottles mainly made by blow-fill seal. And the same is true for the smaller container forms like vials and plastic vials that are also made by blow-fill seal technology. Coming to more specialized container forms like pre-filled syringes can be offered. Syringes can have volumes from 0.5 milliliter up to 50 milliliter and filling volumes even smaller, so filling volume can start with 0.2 milliliter. And we have also some customized systems, especially with bags. We have multi-chamber bags, pillow bags, some cassette solutions, and irrigating systems. So again, thank you very much for your attention. Open, of course, for any questions for you. And again, please meet us for any questions that you might have later on in Hall A44. Thank you very much.